Okay, so our first section before anything else gets started in mitosis is going to be interphase. Interphase comes right before mitosis, and it's what every cycle has to go through before it can even be ready to perform mitosis. So mitosis or interphase consists of three main sections or steps, and those are all G1, an S phase, and a G2. And then G1 is basically going to be getting the cell ready. Nothing really happens visually in the cell in G1, but it's really just getting ready for the next phase. And there is a restriction point in G1, and that restriction point is basically just the cell has to be grown enough to enable to perform the next steps, and if it's not, it'll be sent back and has to just grow. But if it is allowed to pass this restriction point, then it goes into the next phase, and the next phase is going to be the S phase. And then the S phase is where DNA is synthesized, so like multiplied to a huge amount. And then once it is multiplied in the cell, then the cell moves on to G2. And then after G2, it goes on to mitosis. And then starts the mitosis cycle. As you can see here in this image here, this is what our cell will look like in the interphase. So basically just a normal cell. There's a nucleolus that is starting to form, a nucleus, and a nuclear envelope, and the centrosomes are right at the top of the cell. So this is before anything has even gotten started yet in our cell. And then next we'll be going to the first step of mitosis. Okay, so now we start with the mitosis cycle. We start with this just since the cell is past the G2 phase of interphase, and now it goes into the final part of mitosis. And then the first stage of mitosis is prophase. The cell will look something like this in the prophase state. So it's still going to have all those main parts as the last phase, but they're all starting to do different things. The nuclear wall is starting to dissociate. The chromatid bundles are starting to wind together instead of being free. When they wind together, they later on become our chromosomes, but that's not for a few more steps. They all wind around the centromeres, which is just the main little ball in the middle. And the centrosomes are still outside, but they're starting to move to the opposite poles of the cell. These centrosomes are starting to have mitotic spindles starting to grow on them, and then these will later become our kinetochores and microtubules. And then the chromatids are slowly starting to become pairs, but not fully all the way there yet. And yeah. Okay, after the prophase is finished, the cell will then move into the next phase, which is called prometaphase. So prometaphase is where the chromatin pairs that were in the last one are now going to be fully condensed into actual chromosomes. And the identical sister chromosomes that will be made from those condensed chromatin pairs are going to become attached at a point called the centromere of each one. And the mitotic spindles are going to be around the sides of the centrosomes before in the last one are now going to be fully developed into the kinetochore microtubules. And these microtubules will later connect the kinetochores with the centrosomes, but that's for a later step. And now the nuclear envelope is fully broken down, and the centrosomes are on opposite poles of each other, which will later help for the separation. But that's in a much later step. And this is what the cell will look like at the prometaphase step. So as you can see, there's all the different chromosomes, and the microtubules are all across the sides holding on to these chromosomes. And next we will move into the metaphase step. So now that the prometaphase has been completed, the cell is now going to move on to the next phase, and this next phase is going to be called the metaphase. So in the metaphase, the main process the cell is going to go through is really nothing too crazy other than all the chromosomes that are now formed and held by the kinetochore microtubules are going to be lined up on a metaphase plate. This is what that'll look like in the cell right here. And as you can see, it's like the centromeres are on each side and then have all the microtubule strands holding all of them across, and the metaphase plate is right down the middle of the cell, and then all those chromosomes are going to be lined up right there. And then this stage, those chromosomes will get ready to are the most condensed that they'll be in the entire process, and they'll be then getting ready to split apart in the next step. Before the cell actually moves on to the next phase called anaphase, it has to undergo a change called a restriction point, which is also in this case called a CDK point. And this whole process basically is how the cell gets the chromosomes ready to separate by getting rid of the cohesin. So the first step in this process occurs in the prometaphase, where most of the cohesin is removed, except for the cohesin right on the middle of the centromere. There's going to be pictures of all this right here. So the first step is going to have all of it together. Second step is going to have it mostly removed, except for all the cohesin on the centromere. And the last step is after metaphase, where a complex of cyclin CDK activates the APC, which is then going to activate separase, and then the separase is what gets the rest of the cohesin off the centromere and allows the two chromosomes to split apart and then allow the next phase to happen. After the restricting point, the cell can now move into anaphase step, and due to the restricting points process of removing the cohesin so the chromosomes can separate, that's basically what's going to happen in this entire anaphase step. So the metaphase will be finished and the restriction point will be finished, so the cell will go into anaphase. Anaphase is basically where the chromosomes will now pull apart into daughter chromosomes, and then they'll be pulled towards a certain pole on either side of the cell, and then that'll separate the two, and then that's going to look something like this in the picture here. And as you can see, so now the chromosomes are separated onto each side. There's an even amount on both sides. And then next, right after this, will be the last two steps of telophase and cytokinesis. So, second to last step is going to be called telophase, and it's going to come right after anaphase. So in anaphase, the chromosomes are all split apart and broken onto even sides on each side or each pole of the cell. And then now in telophase is where the cell is going to get ready to start breaking apart into two daughter cells. And so the nuclear envelopes are going to start forming again. The cell is going to look something like this here in this picture here. So you can see there's chromosomes, and then there's going to be a nuclear wall starting to form again. And that's going to be starting to look pretty similar to what we started off with in interphase, except now there's two. So yeah, that's the whole point of the last step, is that there was two. They all got pulled apart, and now it's starting to form into two new cells. And then the cell will start to split into two, but won't fully split into two until the next step called cytokinesis. Okay, so our final step of the mitosis cycle is going to be called cytokinesis, and you may guess by now that cytokinesis is just where the one cell separates into two daughter cells. This is allowed because of the last step making basically two cells in that one. So there's two nuclear envelopes and then one centromere on each side, or each pole, each side of the cell. And then with cytokinesis, those two will start to separate. The cell is going to now look something like this picture here. So as you can see, there's now what's called a cleavage furrow. So where they're starting to separate, it's going to make like a little ball, and this little spot here is going to be called the cleavage furrow. And then so the nuclear envelopes are almost fully reformed now, and there's a reforming nucleolus, and it's basically starting to look like two of what we started with in interphase, which is what makes sense for our process of mitosis, of just replicating another cell and making a new one. And yeah, that's all. So our cell will now be split into two cells, and then that's how mitosis works. Thank you.